I'm here once again this week with Bonny Specker and Natalie Thiex from South Dakota State University, who are going to give us an update on COVID-19. Today, Natalie is going to talk about the efficacy of the vaccine, and Bonny is going to give us an update on COVID-19 in our region, in the state, in the nation, and in the world. Now, Natalie, what do you have for us today?
So this past week on Friday evening, the FDA approved with an emergency use authorization an RNA vaccine from Pfizer. And so I wanted to talk about how vaccines work, how this vaccine works, and the data that Pfizer presented to FDA to show that it's effective. Usually when we talk about vaccines, we talk about safety and efficacy, or how well it works, but that's kind of a lot to talk about for one time. So today we'll be just talking about the effectiveness of the vaccine and how that was determined. So before we get started with the details of the vaccine, I wanted to show this cartoon, which kind of shows how SARS-CoV-2, the virus that causes COVID-19, infects cells. This up here in the corner is a cartoon diagram of the coronavirus. And you can see that it binds to receptors on the cell surface and gets internalized into the cell. Below this double line here is representing the cell, a human cell that would be infected by the coronavirus. And after it infects the cell, it can copy itself or make new versions of itself that would go on to infect other cells in the body or to infect other people. And so if we want to both protect ourselves and those around us, one way to stop the coronavirus would be to stop it from getting inside our cells. And that's basically what vaccines help our bodies do. So just briefly, a vaccine can induce our immune system to make antibodies, and those antibodies can prevent the virus, in this case, the COVID-19 virus, from being able to attach to our cells and infect them. So that can both keep us healthy and keep us from infecting other people, in theory. This is a cartoon diagram, a little bit blown up detail of the coronavirus or COVID-19, SARS-CoV-2, the virus that causes COVID-19. And one of the things that I wanted to highlight about this virus is the protein on its surface called spike. So it's represented by these pink spikes that are coming off of the surface of the virus. And this protein is specifically what binds to our cells and allows it to invade our cells. And what we want to do is have our immune system make antibodies or proteins that are made by our body that can stick to this spike protein and prevent spike from being able to help the virus get into our cells. So almost all of the vaccines that are out there, and there are dozens of them in formulation right now in different phases of trials, almost all of them induce the body to make antibodies or proteins that recognize spike. And in this cartoon here, the antibodies are the blue, and the red is the spike coming off of the virus. And what you can see is that when the virus is covered in antibodies, it no longer can interact with our cells and get inside of it. So the antibodies prevent the virus from being able to bind to this cell, and from then, if it can't bind to the cell, it can't get inside of it, and if it can't get inside of it, it can't make us sick, and it can't replicate and go on to infect other people. So that's just a really brief overview of the theory of how antibodies can be induced by vaccines, and they're proteins of our immune system that can prevent our cells from getting infected by the virus. So there have been two current vaccines that have been in phase 3 clinical trials that have enough data to be able to have a preliminary determination of their effectiveness or efficacy. One is the Pfizer-BioNTech vaccine. That's the one that was approved on Friday by the FDA with an emergency use authorization. The other one is by a company called Moderna. And so they're a little bit unique. We've never had an RNA vaccine before. And so I want to explain a little bit what makes these RNA vaccines different from the other types of vaccines that we've had in the past. But basically, the idea here is that we take the spike protein off of the virus, and we actually use the instructions from the genome of the virus in order to get our own cells to make the protein that then elicits the immune response. Our body makes the antibodies, and that's what will circulate in our blood. And when we do encounter the virus out in the wild, that these antibodies that are circulating in our blood that were induced by the vaccine would prevent us from, even if the virus got inside our body, it would prevent it from making us sick. And I wanted to give a little bit more detailed version of that. So normally in the vaccines that we've had prior to this, in at least some of them, we could take the spike protein itself and we could make it in the laboratory and formulate it with the vaccine, inject it into people, and that protein, that small part of the virus, would induce our body to make an immune response. And what's different about these is instead of making the protein itself and injecting that into people, we're going to give the instructions for the protein to our body. So the deal with these proteins is they're very expensive to make in the lab or in bioreactors, and they take a long time. But it turns out that making RNA in the lab is cheap and can be done very quickly. And so that's why we see that the two first vaccines that have been finished or come to the point of the phase 3 clinical trial are RNA vaccines. And I just wanted to give this quick explanation of what DNA, RNA, and proteins are. So DNA is the instructions for how to make a protein. And RNA, we call it the message. It's kind of like DNA, and it takes the instructions from the DNA in the nucleus to the part of the cell where the proteins are synthesized. And so we can just, instead of giving our bodies the spike protein to cause an immune reaction, we can give our body the mRNA for the spike protein, or basically the instructions for the spike protein. So our body will make the spike protein, and our immune system will see it and know to make antibodies that will protect us against the COVID-19 virus. So this is the timeline for the randomized control trial that is the study that is being used to determine the vaccine safety and efficacy. And like I mentioned, we don't have time to talk about safety today, so we'll just talk about efficacy or effectiveness today, and we can talk about vaccine safety next time. But basically, this represents the timeline of this trial. So at the beginning of the trial, so participants are recruited, and there was over 40,000 participants recruited in this Pfizer RNA vaccine trial. And after the participants are recruited, they're randomized to either the placebo group or the real vaccine group. And those participants are given the original dose, and then 21 days later, they're given the second dose. And there's a little bit of leeway, so it's 21 days plus or minus 7 days. And then after that, participants are monitored for up to 2 years for symptoms of COVID-19. So basically the way it works is if you were a participant in the trial and you had any of these COVID-19 symptoms like fever, cough, shortness of breath, muscle pain, loss of taste or smell, then you would inform the study managers, and then within 4 days of the onset of symptoms, be tested with the RT-PCR test for COVID-19. And then so both the people who got the real vaccine as well as the placebo would do this. And then if their RT-PCR test was positive, then they would be categorized as having acquired COVID-19 or having COVID-19 disease. And if they don't have symptoms or they have a negative test, then they would be categorized as not having COVID-19. So one of the things to note from this study design and from these trials as they're currently being run is the outcome is disease. outcome's not necessarily infection. So people could be infected with COVID-19, but if they don't develop symptoms and they're not tested or they don't test positive, they won't be categorized as having the disease. So as of the data that was considered by the FDA last week, the data was collected up through November 14th. And at that point in time, the median for time in the trial was 2 months of follow-up from when the original dose was given, if I recall correctly. So that's the very earliest stage that we could consider this data. And the participants will be continued to be followed for some time after this. So this table shows the demographic characteristics of the participants in the trial. And basically what it shows is that there's about 20,000 participants who got both doses of the vaccine in both the vaccine group and the placebo group. And the participants were randomized to the vaccine or placebo group based on different demographic characteristics like gender or sex, age, race, and whether or not they have any of the comorbidities that we know can make people more susceptible to COVID-19, and also whether or not they're categorized as obese. So this is just showing the number of people in each category and the percentage of the total. So for example, it's about, of the females in the study, half were in the vaccine group and half were in the control group. This table here is showing that there are equal number of participants aged 16 to 55 in the vaccine group and the placebo group. I have a reference here at the bottom of this data. These tables and graphs that are coming up come from a report published by the FDA that was written by Pfizer, the company that was running this vaccine trial. This table is showing that the RNA vaccine was effective in preventing COVID-19 disease across age groups following two doses of the vaccine. So basically, by the time that 2 months of follow-up were completed for the participants in this trial, a total of 170 of them out of the original 40,000 plus that were recruited developed COVID-19 disease. And of those 170, 8 of them were in the vaccine group and 162 of them were in the placebo group. And this is a vaccine efficacy of 95%. And I just put down here the equation for how vaccine efficacy is calculated. We don't need to go over it, but in case anybody's interested, that's what it is. And then in addition to looking at the data for all participants, it was broken down by participants who were younger than 55 years or those who were older than 55 years. And you can see that the efficacy was pretty similar in both groups. So 5 people under age 55 who had the vaccine developed COVID-19 disease, whereas 114 who didn't have the disease didn't have it, and so forth. The next question that people wanted to ask after they said, does the vaccine help prevent COVID-19 disease, is, does the vaccine help prevent severe COVID-19 disease? And so the outcomes for severe disease are things like oxygen saturation below 93%, or hospitalization or death. And there were only 4 cases of severe COVID-19 disease in the study population up through November 14th. So it's a very low number of cases. And of those, 1 of them was in the vaccine group and 3 of them were in the placebo. And in the report that's linked here, it describes exactly what the symptoms of these 4 people were. But I can tell you that this person who's in the vaccine group who was categorized as having severe disease was never hospitalized, but they had at one point had a oxygen saturation of 93%. So that categorized them as having severe disease. So we have to follow up longer to determine if this vaccine helped prevent severe disease. And then the other question was, what if the participants only get 1 dose? Will they be protected? And then also, how long does the immunity to COVID-19 last, or how long does the protection from the vaccine last? So basically what this graph is showing is on the x-axis is the days after the participants received the first dose of the vaccine or the placebo. And so it starts at 0 and goes out through 119 days. And on the y-axis is the incidence of COVID-19. So the number of cases per number of people per population or per study population. And so what you can see is that in the beginning, the red and the blue line is very similar. So for the first 10 or 11 days, the number of COVID-19 cases in the vaccine group was very similar to the number in the placebo group. But then you can see that as the cases continue to go up over time in the placebo group, they level off for the vaccine group. So this shows us that even after one dose of vaccine, there's some protection. Now, most of these people in this group got the second dose on day 21, but some of them didn't, and they were included in this analysis. In the previous slides I showed, if a person didn't get the second dose of vaccine, they were excluded from the analysis. And the other thing you can see is that there is no change really in the slope of this curve over time to suggest that there's any kind of waning immunity and an increase in cases after a certain amount of time. But this is a very, very short time to follow up for this current trial. So we would like to see this extended for many more months to be able to conclusively draw the conclusion that there is a long-lasting immunity from this vaccine. But I'll just throw one thing out there. There's kind of an ethical consideration at this point in time. So the whole thing with vaccines is there's always a risk-benefit analysis, and we have to weigh both risks and benefits to make the decision. For people who volunteered to participate in this vaccine trial, we now know that there's really good evidence of protection from COVID-19. So they may want to, people who are in the placebo arm may want to become vaccinated with the vaccine. And so that's something that our society is going to have to consider of whether or not we're going to make these guys stay unvaccinated for the next 2 years to finish the trial, or whether or not they will be able to be eligible to receive the vaccine. But there are people who may drop out of the study if they want to get vaccinated.
Well, thank you so much, Natalie. I look forward to seeing your next presentation about the safety of the vaccinations. And thank you for giving us an update on the status of what's going on with vaccinations right now. Now, Bonnie, over to you. Do you have an update for us on what's been going on with COVID over the last week?
Yes, I do. I have a couple other slides in addition to the regular ones as well. And I'd like to just mention this following Natalie's great talk on vaccines. In Brookings County, as of December 13th, about 7.8% of Brookings County residents have tested positive for the SARS-CoV-2 virus. If you assume that only half the infected people have been tested, you could say that 16% of the population has been infected. But another way of looking at that is that 84% are still susceptible. And the virus is going to run through that population, or this population, until 60 to 80% of the population is immune. That's the whole idea behind herd immunity. And the consensus right now is pretty much 70% of the population needs to be immune. And just a reminder to everyone that you get immunity either from being infected or actually getting the vaccine. And that if we do that through infection, it's going to result in many deaths. In other words, I like this little summary. There's a lot of wood out there to burn, and if we don't keep the fire tamped down, we're going to have a forest fire. So we need to prevent those forest fires by continuing to wear masks, socially isolating, and practicing good hygiene. But as far as the county, we still are in substantial spread. There have been a total of 2,721 cases. We are dropping a little bit on cases, 127 cases this week compared to 165 in the previous week. As of yesterday, there were 324 active cases. The Sunday before, it was 438. 92 people have been hospitalized since the beginning of this outbreak. We had 7 new hospital admissions this week compared to 6 in the previous week. Brookings County has seen 24 total deaths. 4 of those were this week compared to 4 in the previous week. Our test positivity has dropped some. It's now 28% if you have total people tested as the denominator, compared to 10% if you look at it as among tests that were done. And just a reminder, the WHO, the World Health Organization, recommends that these percents be less than 5. So I wanted to show this slide and show people where we need to be in order to get below substantial spread. Substantial spread is defined as 100 cases per week based on 100,000 population. And that comes out to having less than 35 cases in Brookings County in a 1-week period, which comes out to roughly 5 cases per day. So we need to get this running average, this little yellow line, down below the orange in order to get out of that substantial spread. So we're getting there, but we're not quite there yet. I also wanted to show the rankings as of yesterday. We were last week, Brookings County was #1 in all of these measures, the cases per day over the last 7 days, total cases per 100,000. This is comparing the 10 counties with the most populous cities, and it's ranking them in terms of the lowest rate to the highest. So Brookings actually had the lowest rate of cases and total deaths last Sunday. This Sunday, we dropped down to #3 in the for cases in the last week, and both Beadle and Davison County moved ahead of us. And you can see that they have some of the worst cases of these 10 counties. They have ranks 9 and 10 if you look at the total number of cases per 100,000. And both of these counties did implement mask mandates in the middle of November, which has really dropped these numbers. So that's great to hear. We still have the lowest number of total cases per 100,000 population, and we also have the lowest total deaths per 100,000. As far as South Dakota goes, we have 91,038 cases, 5,037 cases this week compared to a little over 6,000 the previous week. So that's dropping, and you can see that over here. Yesterday, we had 12,747 active cases compared to 16,432 last Sunday. There have been a little bit over 5,000 people that have been hospitalized, 339 admissions this week compared to 367 in the previous week. Yesterday, there were 436 people hospitalized with 82 people in the ICU. And there have been 1,259 deaths. 313 of those have been in December. 149 deaths this week compared to 167 in the previous week. Our test positivity is still quite high, both among people tested as well as among tests done, but it has dropped just slightly compared to the previous week. This is the K-12 and higher ed data, and here we're looking at the slopes of the lines, which is giving us an indication of the rate of increase from 1 week to the next. We would really like to see those slopes move down, and there really has not been a lot of change, at least in the K-12. It's increasing at about the same rate. You could say that's better than it increasing more. You can see here there was a period where there was a fairly rapid increase, and this has kind of slowed down. In the US, we hit a lot of milestones this week. 16.5 million cases, 1.56 million new ones this week alone, compared to 1.37 in the previous week. We've gone over 300,000 deaths. There are 17,000 deaths this week compared to 15,000 in the week before. So we've now exceeded this, and there have been a couple days where we've exceeded 3,000 deaths per day. So that's similar to like a 9/11 event every day. So just to give an idea of the magnitude of that. The US also, the percent of the world cases that are from the US is actually going up, and we now represent 23% of all the cases in the world. So worldwide, we have 72.1 million cases, 5.25 million this week, compared to 4.28 million in the previous week. You can see here that there's been a pretty rapid increase in the last month. Deaths, we have 1.61 million deaths, 77,083 this week compared to 76,284 in the previous week. So you can see that these are also increasing and are at record level highs. There were a couple changes with the travel restrictions this week. Minnesota was removed. There's no more travel restrictions there, while Oregon was added. And, you know, again, if anyone's thinking of traveling, you might want to check that website. It is open to people who don't have a subscription. You can get on there, and there's links to all the different states that you might be interested in traveling to. So we need to do what we have to keep this under control, and I think people know what that is by now. You know, wear masks, socially distance, and practice good hygiene, and we just need to keep it up and do it. So that's all for this week.
Well, thank you, Bonnie, and thank you so much, Natalie, for putting together those presentations for this week. And next week, Natalie will be talking about vaccine safety, and then we'll have another update from Bonnie as well. If you have a question about epidemiology that you would like them to answer, or contact us at the email address listed in the description of this video, and we will get those questions answered for you. Until next week, keep doing what you need to do and stay safe. Thank you.